Hi everybody, welcome to chapter 3. In this chapter we're going to be looking at microscopy, which is how we're able to see these microscopic structures. And then we're also going to talk about the cell structures themselves. So using the microscope we're able to see structures that are smaller than our normal naked eye could see. But even when we're looking at bacteria or other types of structures that are super small, a lot of times we have to stain them to make them look different from the background. And so Hans Christian Johann Graham was working at in a hospital, in a morgue, and he was trying to identify the causative agent of pneumonia. And so he was developing a stain technique to stain bacteria. And in one of the methods that he did, which we'll learn in this chapter, is known as the Graham stain. Some of the cells retained one of the dyes, the primary dye, and the others retained a secondary dye. And this was the basis for what we now call the Graham stain. And so he actually was able to separate organisms based on the chemistry of the cell wall, so what the cell wall is made of. And so we know now that there are two major groups of bacteria, gram-positive and gram-negative. So the invention of the microscope led us to identify two major groupings of cells, the prokaryotic cells and the eukaryotic cells. Prokaryotic cells are always smaller in size, which means they have a larger surface area to volume ratio, which allows them to take nutrients in much faster. But they're also more prone to predation, parasites, as well as being outcompeted. So microscopes weren't a common standard in labs until the mid-1600s, when the first microscopes were actually produced, the ones that were actually produced and described in research or published. So light microscopes, which are the most common ones, they can magnify items up to 1,000 times their actual size. And this is what we use in our labs. Electron microscopes can magnify up to 100,000 times or more. And they were invented in the 1930s. And then we have increased their function. In the 1980s, we produced scanning probe microscopes. They can identify individual atoms on the surface of a cell. So, I mean, super strong microscopes. We're going to focus on these microscopes because these are what we use in our labs. So how they work: light passes through the specimen and then through a series of magnifying lenses. This allows us to see the structure. The microscope that we actually use is known as a bright field microscope, which produces a dark image on a bright background. We have these microscopes in our labs at Sandburg College. Three concepts that we will discuss while talking about these microscopes are magnification, so the increase in size of whatever we're looking at; resolution, the ability to distinguish two objects that are close together as separate objects; and contrast, so a difference in either color intensity within the object or color from the the object itself and the background to be able to see that structure. So we're talking about magnification. This is a typical compound microscope. This is what we have. We have binocular microscopes, so that means two ocular lenses. Your ocular lenses magnify the image 10 times whatever the size is. So if the image is 1 millimeter, then when you look under the ocular lenses alone, you would see it at 10 millimeters because it magnifies by 10 times. The stage is where we place our slides. These little, what do we call graspers? These are what hold the slide in place. The objective lens, so this is a revolving nose piece here, and it contains 4 different objective lenses. The 4 objective lenses magnify the smallest, which is known as scanning, is 4 times magnified. Then we have the low power, which is 10 times magnified. We have high power, which is also known as high dry, because you do not get it wet with oil ever, is 40 times magnified. And then the highest power is your oil immersion. This one has to have oil, so you have to, it's going to be a wet lens, is 100 times magnified. So you're going to multiply the ocular lens by the objective lens to see how large that object is going to look under the microscope. There's a condenser. The condenser helps to focus the light on the specimen, but it does not magnify anything. Here is your light source. Over here we have the large knob is called the coarse focus. The tiny knob here is called the fine focus. Coarse focus moves the stage up and down. Fine focus is going to tweak the image. So we use coarse focus on lowest power, so scanning, and low power, but we use fine focus on all 4, scanning, low, high dry, and oil. Okay. You have an iris right here that controls the amount of light that is let in, so you can turn the amount of light down, or you can open it up. If the image is going to be very, very clear, you might want to close the lens so you can see it a little bit better. Resolution. So resolution is the ability to distinguish 2 objects that are very close together as separate. And the resolution depends on the type of lens that you're looking at, as well as the magnification and the light waves. So typical resolution of a light microscope is about 0.2 micrometers. We can increase resolution by using immersion oil. So when we are looking at something under the oil immersion lens, we add a drop of immersion oil to the slide, and that decreases the refraction. So refraction is the light rays scattering. So we prevent that refraction with the oil so that the light is going to go through the light source, through the object, and then to the lens. And then the last is contrast. So contrast determines how easy we can see the cells that we're looking at, or the structure. So transparent bacteria do not have contrast, and so we stain them to produce a contrast. But when we stain bacteria, we typically kill them. There are some microscopes that you can use that produce a contrast, and you don't have to stain them. We don't use them in our lab because we don't have them, but there are microscopes that do that. All right, I'm going to stop here and I'll get into all the other microscopes. So we talked about the bright field microscope is the one that we use. That's the one that I showed you a picture of. Bright field microscopes produce a bright or an image on a bright background. The next microscopes we're going to talk about are also light microscopes, but they differ slightly from the bright field microscope, okay? So I'll talk to you about those microscopes in the next video. Bye.